Basically, biologics, biopharmaceuticals are any type of therapeutic that are semi-synthesized or synthesized or extracted from biological sources. They include proteins, peptides, nucleic acids, and in contrast to small molecules, they have secondary, tertiary, and quaternary structures. So they're composed of amino acids, which can take on different charge states, which have different hydrophobicity. So they're much, much more complex because of this. Additionally, they can have post-translational modifications, right? So anything like glycosylation or oxidation or reduction that can change the exact three-dimensional structure of a biologic. And the exact three-dimensional structure of a biologic is very important because changes in the 3D structure can have changes for affinity and have different immunogenicity and high-impact safety risks. So one of the things, does this guy work? Okay, so an analogy that's often drawn is that small molecules are relatively easy to synthesize. They're relatively easy to characterize. You can pretty much see everything, so they're kind of like a bicycle. Whereas if you go up to peptides and RNAs, they're a little bit more complex. They're kind of like a car. And a monoclonal antibody, which has a molecular weight of around 150,000, is kind of like a fighter jet, right? So that's the challenge here chromatographically, is these things are way, way more complex. So why are we interested in biopharmaceuticals? Well, in 2014, it was a $175 billion market, right? 50% of pretty much all new drugs are predicted to be biopharmaceuticals. In 2014, biologics accounted for more than 31% of total drug spending. So that was up 3.3% from 2013. And right now, there's over 200 biopharmaceuticals on the market, and they're achieving blockbuster status. So that means that they're grossing over $100 billion a year in revenue for the developing company. And of these 200 biopharmaceuticals, a lot of them are now coming off patent. So generics are coming in, and we're developing biosimilars and biobetters. Okay, so here are the top 10 pharmaceuticals in 2016. Only one of them is a small molecule, Revlimid. It's a thalidomide analog for the treatment of myeloma. All other 9 are biologics, and the ones that are actually highlighted in yellow, these are all monoclonal antibodies. So close to home, you can see Humira, developed by AbbVie, is the top monoclonal antibody drug in 2016. Okay, so now let's kind of look at the monoclonal antibody market. So in 2011, it was valued at $45.1 billion, and they projected that it would reach up to $67 billion by 2015, but it actually grew up to $85.4 billion in 2015, right? So that's pretty huge. And now, by 2024, it should be about $140 billion. So why is it growing so much? And basically, mAbs are a great solution as a therapeutic for the rising incidence of cancer and chronic disease. And as chromatographers now, we're being faced with a variety of new and challenging methods for analysis, and that's what I'm going to talk about today. So who are the players in biopharma and biotherapeutic development? Well, here's the drug discovery pipeline, right? You have your discovery stage, your development stage, and your commercialization stage. Big biopharma is involved in all 3 stages of this process, where you'll see biotechs and startups being involved in discovery and development. Nonprofits like academia are just mostly in discovery. And then you have your contract researchers, or contract research organizations in discovery and development, and contract manufacturing, kind of taking over ending development and commercialization. So basically, if you work in any of these, you're going to start to see biotherapeutics, right? If you haven't already, which you likely have. All right. And then even to drive the point home a little bit further, large pharma is definitely moving towards biopharma, as I've mentioned, but here is basically the change in revenue. So the change in percent revenue from pharma to biopharma between 2000 and 2012, and then the total percent revenue for some of the big pharma companies. And just if we look at Pfizer and AbbVie, you can see Pfizer grew 29%, AbbVie grew 52%, and this is in 2012, so way bigger. All right, so now I've given an extensive background as to why this is important, but let's go over and do a brief introduction of what monoclonal antibodies are, their structure, the type of post-translational modifications that can occur, and then let's look at how we're going to analyze them chromatographically. So the three ways I'm going to talk about are aggregate analysis by size exclusion, intact analysis by reverse phase and peptide mapping, and then charge variant characterization by ion exchange using pH gradients and salt gradients. Okay, so first, what is an antibody? An antibody is a protein produced by beta lymphocytes, so white blood cells, and basically its purpose is to identify and neutralize harmful foreign objects in your body. Monoclonal antibodies are antibodies that are identical, so identical proteins, because they are produced by clones of a single parent cell. So you take an immortal tumor cell, you fuse it with a beta lymphocyte that produces a certain type of antibody, you form a hybridoma. Those hybridomas produce monoclonal antibodies. So these aren't the type of antibodies that are produced in your body, right? We make them specifically to be therapeutics. As opposed to polyclonal antibodies, which are antibodies that are derived from a variety of different types of immune cells and have a variety of different antigen recognition. There's also isotypes of antibodies, so not all antibodies look like a Y, right? And they're characterized according to their heavy chain constant domains. So you've got IgG, IgA, IgM, IgD, IgE. Basically, the big letters here stand for their heavy chains. Okay, so what is a heavy chain? What does an antibody look like? Here we have a structure, cartoon structure of IgG1, and we have an X-ray crystal structure of IgG1. So basically what you have here in an IgG is you have two identically sequenced heavy chains that are connected by two interchain disulfide bonds. And then you have two identically sequenced light chains that are connected to the heavy chains by these disulfide bonds just above what is called the hinge region. There's two variable regions of an antibody that occur here, right? So you have a variable region of the heavy chain, the variable region of the light chain, and that terminates in the antigen binding site. Okay, so these are the two variable regions at the tip of the Y. The other thing to notice here is that at the hinge region of the IgG, you have a papain cleavage site. So you can take a papain protease, you could come in here and cut this thing into three fragments. The FC fragment, which is the lower fragment, and then two FAB fragments. So two antigen binding fragments. Okay, and now what type of post-translational modifications happen in antibodies? Well, if we look over here on the crystal structure, you can see you can get asparagine deamidation, asparagine glycosylation, you can get aspartic acid isomerization variants, you can get serine glycosylation, threonine glycosylation, and you can get oxidation of basically any cysteine-containing amino acid, right? So you're going to change the oxidation state of disulfides in cysteine, or oxidize histidine or methionine, right? So these are the types of post-translational modifications that can happen that affect the immunogenicity of antibodies, right? So post-translational modifications kind of manifest themselves in two ways. You either get acidic variants or you get basic variants when you're talking about antibodies. So the acidic variants come from silation or deamidation or C-terminal lysine cleavage. And the basic variants can come from succinimide formation, oxidation of tryptophan, histidine, lysine, cysteine, or methionine. You can get disulfide reduction. You get acylation or C-terminal lysine cleavage. So all of these things are things that occur after you have your therapeutic that change its efficacy. Okay, and because of that, we can see that characterization of mAbs can be a huge challenge, right? So you change one little thing and you have a completely different therapeutic, essentially, that gives rise to off-target effects. So like I mentioned, the biologic activity of the therapeutic efficacy depends on the exact three-dimensional structure, which is affected by these post-translational modifications. So what has kind of happened now is the International Conference on Harmonization Guideline Q8 has set forth a guideline of physical, chemical, biological, and microbiological properties that need to be kept within a certain limit, right? So that you have a safe an effective drug. And these guidelines include purity and process guidelines, product variant guidelines, and drug product attribute guidelines. So mostly I'm going to talk about the product variants, so how we're going to use HPLC to look at those, aggregation, conformation, see what states the C-terminal lysines are in, see if there's deamidation isoforms. So this is all these product variants are all the things that we use HPLC to analyze. Okay, I'm going to grab my water. Actually, Tammy, can you hand me water? Thank you. I appreciate it. Okay, so like I mentioned, I'm going to talk about 3 different types of chromatography for mAbs. The first thing I'll talk about is size exclusion for aggregate analysis. Then I will talk about reverse phase chromatography for intact mAb analysis and peptide mapping. And then I'm going to talk about ion exchange to determine charge heterogeneity. All right, so let's jump in to size exclusion and look at aggregate analysis. Okay, so why are we doing this? Basically, high molecular weight species, or aggregates, are associated with immunogenicity, right? So we need to characterize if they're present or not. We need to separate, and size exclusion functions by separating these molecules based on their size, right? Or hydrodynamic volume. What happens is you exclude them from an inert porous matrix of controlled pore size and separate them by size. So we have a really cute little cartoon here where we're separating a mAb, which has a molecular weight of 150,000, from a protein that has a molecular weight of 66,000, from a small molecule that has a molecular weight of 112 Daltons, right? And what we saw, if it wasn't too rapid, is that when the mAb was traveling through the column, so all 3 of these guys were injected as a single band, right? And as the mAb was traveling through the column, it was excluded from all the pores. So it had a smaller diffusion pathway, essentially, and it came out first. Then you had your protein that was in the middle of sizes, and that came out second. And then the small molecule was able to diffuse in and out of all of the pores, so it came out last. It spent a lot more time on the column. So that's basically how size exclusion works, right? The difference between size exclusion and like reverse phase is that typically you use smaller flow rates, or between 0.3 and 0.4 mils per minute. And you generate a little bit less back pressure because of this. So on a 150 by 4.6 millimeter column, you'll have around 200 bars of back pressure. The exclusion range that you use is going to be based on the pore size of the particle. So for instance, here we have a variety of different particle sizes with a variety of different pore sizes. So a pore size of 145 angstroms gives you an exclusion range between 1,000 and 300,000, whereas with 450, you have an exclusion range of about 15,000 to 1.5 million. More recently, we and other column manufacturers have come out with sub-2 micron particles for size exclusion. These are really great because you can get shortened analysis time and better peak resolution, and you don't actually have to use a UHPLC for these guys. The back pressure generated is, because of the slow flow rate, is not a UHPLC typical back pressure. Okay, so when you're doing size exclusion, what are some typical conditions? First, you want to use non-denaturing mobile phase conditions. You typically run at room temp with an isocratic mobile phase with 0.2 or 0.5 molar sodium chloride or potassium chloride. So I've learned from some of our guys at Phenologics, at our Phenologics lab, that potassium chloride actually does a little bit better of a job because potassium's a bigger ion, so it shields residual siloxanes a little bit better, and you get less kind of secondary ion exchange interactions. You'll want to keep your pH about 1.5 to 2 units away from the PI of your protein, so usually around 6 or 7 if you're talking about a mAb. And then the particles in size exclusion are silica or polymer based with some type of hydrophilic coating. So I kind of mentioned this already. You really want to avoid secondary interactions when you're doing size exclusion because you want to have like a pure mode of size exclusion interaction. So coating the particles with the hydrophilic coating kind of reduces the amount of hydrophobic interactions that you get. And then again, you're using salt to basically shield any residual siloxane interactions. This is an example of size exclusion with Adalimumab. You're using a sub 2 micron 300 angstrom column here. And basically what this is showing is the aggregate has an area percent of 0.16. So a sub 2 micron column gives you the best resolution for this. You probably wouldn't be able to see this on like a 3 or a 5 micron size exclusion column. Okay. Here's an example of size exclusion chromatography with mass spec detection. This is cool for 2 reasons. First, so now because of the rise of biologics, column manufacturers are coming out with new and improved size exclusion columns. So they're a little bit more inert, essentially, than older versions of size exclusion. And what that means is that you don't have to use as much salt. And what that means is that you can do mass spec detection. So that's the first reason this is kind of a cool example. The second reason is here we have 6 types of proteins, model proteins, okay? The biggest one is alcohol dehydrogenase. It has a molecular weight of 150 kD. The smallest one is cytochrome C. It has a molecular weight of 12 kD. So we have 2 different size exclusion columns here. The one on the left has a pore size of 150 and an exclusion range of 1,000 to 450 kD. The one on the right has a pore size of 300 and the exclusion range is 10 kD to 700 kD. So all of these proteins fit within both of the exclusion ranges of these columns. But you can see if we just look at the ADH, the alcohol dehydrogenase, that the 300 angstrom column does a much better job resolving. And if you look at the smallest protein, the cytochrome C, the 150 angstrom column does a much better job of resolving. So there's a couple things that you've got to kind of think about when you're doing size exclusion. Even though all of these proteins fit in the exclusion ranges of both, one is going to be better for one and the other is going to be better for the other. So I talked about this a little bit, but this is a cool example. So you want to minimize as much as possible secondary interactions that you get with the column, right? You want to have purely size exclusion interactions. So how you can do this, how you can modulate this, is by the concentration of salt that you use in your size exclusion buffer, okay? So here we have an example of some biosimilar. We're running it on a sub 2 micron 300 angstrom size exclusion column. The dimension is 300 by 4.6. And the buffer we're running is 50 millimolar potassium phosphate. And now we're varying the amount of potassium chloride. We're varying the concentration of potassium chloride. So now if we look down at the bottom, we have 0 potassium chloride, right? And you see this peak that is kind of blobby and it's tailing, right? And the reason that this peak is tailing is because now your protein is participating in ion exchange interactions with the silica phase, right? No matter what you do, you're going to have residual siloxanes. Those siloxanes are going to interact via ion exchange with your protein. That is a strong and slow interaction. That's going to have different kinetics. You're going to lead to peak tailing, okay? So now as you go up in salt, let's go all the way up to 300 millimolar potassium chloride. Now you see bad peak shape again, right? Why? That's now because you've increased the ionic strength of your mobile phase so much that your protein's trying to basically glom onto anything hydrophobic, right? It's either glomming onto the phase or it's aggregating and glomming onto itself. So you really kind of have to find the sweet spot when it comes to the amount of salt that you're adding. And for this particular biosimilar, that sweet spot's around 200 or 250 millimolar. Okay. So now we've talked about size exclusion, we've talked about mAbs. Let's get into hydrophobicity and reverse phase chromatography for intact analysis of proteins, and let's also talk a little bit about peptide mapping. Okay, so reverse phase is great because there's a ton of different applications you can use it for, right? You can use reverse phase for full-size intact proteins and mAbs. You can use it for the FAB and the FC fragments. You can use it for reduced heavy and light chain analysis. You can also use it for peptide maps. It's a very, very well understood hydrophobic mechanism. Everybody's basically familiar with it already, right? So the concept is pretty much easy to understand. Column manufacturers have been developing reverse phases for years, so there's a ton of different bonded reverse phases in a ton of different dimensions with a ton of different particle sizes and particle types. And the other great thing about it is that you can use mobile phases that are compatible with mass spec detection, okay? Obviously, reverse phase chromatography uses hydrophobic differences to assess differences in your process formulation development samples, any sample along the way with your protein in it. But these hydrophobic differences that reverse phase utilizes can tease apart differences in hydrophobicity from post-translational modifications, right? So methionine oxidation, glycosylation, reduced thiols, all of these things can be teased apart using reverse phase. Okay, I've said a bunch of great stuff about it, how we know a lot about it and have been using it for years. But one thing, a caveat to this, is that us and other column manufacturers have developed specific phases for proteins and mAbs for a reason. You're going to want to use a phase that has low silanophilic silica. Typically, you're going to want shorter alkyl chains if you're doing intact analysis. You want to use a non-porous, a superficially porous, or a wide pore material. The kinetics of mass transfer, the kinetics of diffusion for a protein are much slower, right? They're bigger. The other thing is that you need bigger pore sizes because proteins are bigger. So there are particular products that you're going to want to use for reverse phase and proteins. And then the other thing that is now on the market are more efficient sub-2 and sub-3 micron particles that are specifically made for biologics. Okay, so here's an example of why you want to use a specific type of particle for intact analysis. So we're looking at an intact protein, and we're comparing a fully porous particle to this core shell particle. They both have wide pores, but what you can notice is with the fully porous particle, you're getting a very broad peak shape and you're getting pore recovery. This is primarily a consequence of the amount of surface area that's in a fully porous particle, right? So your protein is diffusing into and out of the entire particle, and because of the increased surface area, there is more opportunity for your protein to adsorb to the column, right? Whereas with a core shell or a superficially porous particle, your protein can only diffuse into and out of essentially the fully porous silica that coats the particle. This gives better efficiency for a couple different reasons. The interaction pathways are narrower. Second, with core shell particles, they have a narrower particle size distribution. Basically, with a fully porous particle, these guys are formed by the polymerization of tetraethoxysilane. It's a reaction. There's going to be a distribution of particle sizes. If any column manufacturer sells you a 3 micron particle, there's going to be some that are 5, there's going to be some that are 3. It's a much wider distribution. Whereas with a core shell particle, they're made by grafting successive layers of fully porous silica on top of an inert core, which is basically all the same size, right? So you can pack them more efficiently into a column, and therefore they give you better efficiency in your chromatography. Okay. When you buy a core shell particle specifically for a protein from a manufacturer, it's going to be different than a core shell particle for a small molecule. Basically, you have to optimize the phase to core ratio. So with us, the phase ratio is much smaller. There's less actual porous silica on top of the phase for the protein to diffuse into and out of. And they're also usually made a little bit more stable because for reverse phase protein chromatography, a lot of the times you have to operate at higher temperatures. The offerings that you will typically see for peptide mapping are C18s, and then for intact analysis, you can see C3s, C4s, C8s, stuff like that. All right, so when would you use a C4? Here's an example of PEGylated proteins. So PEGylated proteins, intact PEGylated proteins, can be difficult to analyze because they're pretty hydrophobic, right? So here we have an example of a monopegylated bovine serum albumin protein. and then a variety of PEGylated carbonic anhydrases. And you can see on the C4, you can completely resolve the PEGylated BSA from BSA, and you can also resolve the PEGylated carbonic anhydrases from the non-PEGylated carbonic anhydrase. You need to optimize your gradient a little bit here, but this is an instance where you would use a C4, right? When you need to get all of your protein off the column, when you don't need that much retention. Okay, what is the effect of temperature on protein recovery? So here we have intact Adalimumab on a wide pore core shell C18 column with a typical mobile phase of water and acetonitrile. At 70 degrees, you see a little bit of tailing. If you go up to 80 degrees, you can see that you're getting much better recovery and a little bit less tailing. If you go up to 90 degrees, you get the best chromatography, okay? Typically, heat mitigates secondary interactions. It's not always true, but one of the things that you should vary when you're looking at proteins in reverse phase is temperature. Here's an example where it's not always true. Okay, so we have bevacizumab fragments. We have the FC and FAB fragments with a typical water acetonitrile mobile phase on a C18. And you can see here at 70 degrees, the peaks 2 and 3 are resolved. Peaks 9 and 10 are resolved. Peaks 11 and 12 are becoming resolved. But actually, when you increased your temperature to 90 degrees, you start seeing coelution. So 2 and 3 are coeluted, 9 and 10 are coeluted, 11 and 12 are completely coeluted. So the take-home message here is when you change your temperature, you change your selectivity. You're not going to really know what it's going to do until you do it. You just have to try. But typically, it will give you better chromatography for proteins. Okay, so now let's take a quick detour into peptide mapping. What is peptide mapping? In peptide mapping, you have your intact protein that you're analyzing. You cleave it with a specific protease to generate a variety of peptide fragments. And then you analyze those fragments via reverse phase chromatography. It is a common technique for the characterization of mAbs to determine product identity, to determine product stability. And a lot of the times these experiments are used to determine the identification, localization, and relative quantification of post-translational modifications. When you're doing a peptide mapping, the main thing that you really have to think about is peak capacity. Basically, peak capacity is how many peaks you can get in your chromatogram, right? You want as many peaks as possible because you want to get the best sequence coverage. You want to get as much information as you can about your protein. So you calculate your peak capacity via this equation down here. So the higher the n, which is peak capacity, so it's n equals 1 plus your time gradient divided by your average peak width, right? So basically narrower peaks give better capacity. One of the other things to think about are just to keep in mind when you're doing peptide mapping is typically you'll see gradients of 0.5 to 1% organic per minute. So very kind of shallow gradients. And then a lot of the times you'll see 250 millimeter columns, really long columns with really shallow gradients to get the best sequence coverage. So basically what's recommended is to always use a superficially porous particle for this, right? So here we have a comparison of a core shell 1.7 micron C18 versus another 1.7 micron C18 that is a fully porous particle. And you can see that the core shell particle is going to outperform the fully porous particle, right? It has less surface area, it's more efficient, it's going to give you a better peak count. So you get 22 extra peaks out of the core shell particle than you do out of the fully porous particle. The acid that you use is going to change your chromatography in peptide mapping. So here we have an example of formic acid versus trifluoroacetic acid. So on the left-hand side, we're using trifluoroacetic acid. On the right-hand side, we're using formic acid. We're looking at this phosphorylase B peptide digest. And you can see that TFA gives you much better chromatography, right? You have a better peak capacity. It's ion pairing, it increases your hydrophobicity, it increases your selectivity. But a lot of the times, actually, formic acid is preferred because it's not ion suppressing. So it's just important to keep in mind that the acid that you use is going to give you different chromatography, and it's really what you're trying to achieve. So how do you start a peptide mapping experiment? You start with a scout gradient. So typically what you'll do is you'll have your peptide digest, and you'll start with an initial low percent organic, a low percent B. Typically around 2 to 5%. You'll run 5 to 10 column volumes of that mobile phase. That's to get the early eluting peptides, the really polar peptides, to stick to the column. Then, after you've done that, you're going to run about 0.5 to 1% organic per minute on your slope, up to about 60% organic. And most of your peptides should elute here. Afterwards, you ramp to 95% to get anything else that's stuck on the column off and to re-equilibrate. So then once you've done your scout gradient, you can really zoom in here and figure out what you want to do, analyze, go after the information that you're actually looking for, right? But something to keep in mind is that your elution order will change for peptides as your gradient changes. So here's an example of that. So here, we have a longer peptide and a shorter peptide, and we're using a 0.23% acetonitrile per minute gradient. And typically what you'll see with very slow gradients is that the longer your protein is, the more it's going to retain. However, if you increase your gradient, you can actually see the elution order change, which is what we see here, right? So we've actually flipped these two peptides when we've gone up to 1.3% acetonitrile per minute. So this paper is actually where I got this from, but basically they looked at 47 peptides in total, and they found 19 inverted pairs, right? So even just when you're zooming in on your gradient, keep that in mind if you're changing it. Okay, so finally, we're going to talk about ion exchange chromatography, basically why we use it, what it is. This is more the bulk of the talk here, but basically we're going to talk about ion exchange to determine charge heterogeneity in monoclonal antibodies. Why is ion exchange important? It's used a lot of the times for release and stability specifications for monoclonal antibodies, for quality control, and as now biosimilars and biobetters are coming up, it's a key purity assay to look at charge variance. So it's important. 20% of amino acids in IgG1 are charged. And as we've discussed, a lot of post-translational modifications can make variant forms, right? So these variant forms have alterations in their charge-based content, and that is what's used to essentially tease out differences with ion exchange ligands because they result in different retention profiles. So there's two things that you're using ion exchange for. It can be used analytically for monitoring changes in the product profile, but also it can be used preparatively to isolate a charge variant or a post-translational modification for further characterization and identification. All right, so let's just go over the basics of ion exchange. It uses ionic interactions between your analyte and the stationary phase to stick your analyte to the stationary phase, right? So the stationary phase carries the opposite charge of your analyte, and then once your analyte is bound, you either use a salt gradient or a pH gradient to elute it. Why would you use either one? Well, salt gradients are a little bit more common. They're simple to make. People have salt in their labs. It's easy to control the salt concentration of a buffer. It's easier to control the gradient of a buffer with a salt concentration. But the main drawback of using a salt gradient is that you need to always optimize your buffer conditions, right? For every different mAb, you have to optimize a different condition. Every different protein, you have to optimize a different condition. pH gradients are a little bit more preferred because the separation depends on the isoelectric point of your protein. You have lower salt in your collected fractions, and in many cases, you get better resolution. Okay, so let's take a step back and let's think about the PI of a protein. So the PI, or the isoelectric point of a protein, is the pH at which your protein takes on a neutral charge, right? So as a small molecule chromatographer, as a small molecule organic chemist, I always just think of the PI like the pK of the protein, right? The difference is, as you go down in pH away from the PI, you can add more and more positive charge to your protein, right? So if your PI, or if your pH is way less than your PI, you're going to have a much more positive charge. If you increase your pH above your PI, now your mobile phase is basic, it's going to deprotonate your protein, so you're going to have a negatively charged protein. Here's another way to look at this, right? So here we have the PI or the isoelectric point of your protein. As you get farther away from the isoelectric point on the acidic side, you get more and more positively charged proteins. As you get farther away on the basic side, you get more and more negatively charged proteins, right? So you can use this to your advantage in ion exchange. All right, so now we've talked about what is ion exchange. We've talked about the types of gradients that or the types of buffers and elution profiles that can happen in ion exchange. Let's take a step back and actually look at the stationary phase. The stationary phase in ion exchange is different than size exclusion, and it's different than reverse phase. Basically, ion exchange is a very strong and slow interaction, right? As opposed to like hydrophobic interaction, which is relatively weak and transient. So you want to reduce as much as possible the contribution of diffusion from your particle. So typically what happens with ion exchange then is that you use non-porous particles. You don't want to have any diffusion of your protein into and out of pores because you'll get super broad peaks. So these guys are made by polymerizing divinyl benzene with vinyl benzene chloride. This makes polystyrene divinyl benzene particles. These are super hydrophobic, so typically column manufacturers will coat them with hydrophilic coatings like polyvinyl alcohol, and then on top of that, they will graft their ion exchanger. The caveat, the drawback to ion exchange, because you are using a non-porous particle, is that they have a reduced loading capacity, right? They don't have a lot of surface area, so they have a reduced loading capacity. And you can exceed the loading capacity rapidly, so you'll start to see poor peak shapes. All right, so what are the ion exchangers that are used in ion exchange chromatography? We're going to talk primarily about cation exchange because that's used mostly for mAbs. But usually for a weak cation exchanger, you will see a carboxylate. And for a strong cation exchanger, you'll see a phenylsulfonate. The other thing that you oftentimes see with ion exchange columns is they'll either be made of PEEK or PEEK-lined stainless steel. This is to reduce any interaction of your protein with active sites in metal. Okay, so now let's kind of talk about an ion exchange chromatogram for a monoclonal antibody. What does it look like? Typically, like I mentioned, for monoclonal antibodies, you're going to be doing cation exchange with a carboxylate or a sulfonate cation exchanger. That's because the PI of mAbs is around 7 or 8 typically. These chromatograms have a main peak, which is typically your peak of interest, with minor flanking regions of acidic or basic variants eluting before or after. So acidic variants are going to elute before the main peak because they're less basic. Basic variants are going to elute after the main peak because they have more of a positive charge. You're not going to get a lot of peak resolution specifically for the acidic variants because they come out first. However, when you have termini variants, those are typically well resolved. They're more accessible. The buffers that you use in cation exchange, phosphate and morpholinosulfonic acid are the most common. Phosphate is readily available, so it's used a lot, but it's actually a bad choice because the pH of phosphate changes depending on ionic strength, right? So the more salt you add, the more you're going to change your pH with a phosphate buffer. And in ion exchange, changing the pH is going to change the charge state of your protein, so that's not something you want with a salt gradient, right? So actually MES is a much better buffer for cation exchange. It's got a good mid-range pK, doesn't form coordination complexes, and its pH doesn't really change based on the amount of salt you have. All right. One of the things that you can do when you get your ion exchange column is assess the ion exchange capacity. You can take a well-studied protein. Here we have equine cytochrome C, and run an isocratic method, and your retention will correlate to the charge and the amount of ion exchange, while your peak asymmetry and efficiency is going to give you an indication of how well the column's actually packed. And like I mentioned with cation exchange, here's an example. Your acidic variants are coming out first, right, because they are less positively charged than your peak of interest. So now let's look at salt gradients and how you run them and why they're run. So when you're running a salt gradient, the first thing you want to do is fix the pH of your mobile phase less than that of the PI of your protein. It should be 1.5 to 2 units below the PI of your protein. With a salt gradient, you're going to start with a very low ionic strength, right? So a low concentration of whatever salt you're using. And then you're going to elute your proteins by increasing the ionic strength of your buffer as you go on. Okay? Increasing the ionic strength of your buffer increases the competition between the buffer ions and the bound cation exchanger. And like I mentioned before, salt gradients typically must be tailored for any different protein that you're using, right? You can't just go in and say, this salt gradient worked for this protein, I'm going to try it on this. It has to be tailored for your exact protein. All right, so here's a visual of what I just said. You have a mixture of 3 different proteins here. They have 3 different PIs, but you choose a pH that is below all 3 of the PIs, right? So that means one protein has the most positive charge, represented here by this red protein with 3 positive charges, and one protein has the least amount of positive charge, represented by the green one with this positive charge. You load your mixture of proteins onto your column, and they all stick because you're in a pH that is less than the PI, right? Then, oh, yeah, of course. And because you have a very low concentration of salt, right? So now here, you want to start eluting your proteins based on their charge. So you run a salt gradient, and at low concentrations of salt, the sodium atoms come in and they compete for the phenylsulfonates, right? So you kick off the least positively charged protein first, that's the one that elutes, and then as you increase the salt gradient sufficiently enough, you will elute the second positively charged, and then finally the third positively charged protein. So a chromatogram looks like this, right? The protein with the least amount of positive charge comes out first. The protein with the most amount of positive charge comes out last. Okay. In using a salt gradient, you can still modify your pH so that you can get more or less retention. This is an example of that. So you have a protein that has a PI of 8. If you choose a pH of 6.5, you're only 1.5 units below the PI. So it has less of a positive charge, so it's going to elute pretty early in your run. Whereas with the same protein at pH 4.5, which is 3.5 units below the PI, you're going to have much more retention because the pH is much lower than the PI, right? So you can use pH in a salt gradient to still modify retention. Okay, so now we talked about salt gradients. Let's talk about pH gradients really quick. Again, just like with the salt gradient, you fix the pH of the starting buffer below the PI of the analytes that you're analyzing. For cation exchange, again, it's below the PI. If you're doing anion exchange, it'd be above the PI. You keep the overall salt concentration low because you don't want salt ions competing for interaction with your cation exchanger. And then you elute your proteins by changing the pH of the mobile phase. So in cation exchange, you're going to bring up the pH of the mobile phase, right? As the protein approaches its PI, it changes to a net charge of 0 and it elutes from the column. So here's an example of this. You have 2 monoclonal antibodies. The green one has a PI of 8.2. The blue one has a PI of 6.8. At a pH less than both of their PIs, they both bind to the cation exchanger. Then you increase the pH using a gradient, as it reaches a pH of 6.8, which is the pI of the blue protein that elutes. And then as you increase the pH even more to 8.2, you elute your green monoclonal antibody. Okay, so I hinted that pH gradients are preferred. One of the reasons pH gradients are preferred is because you can actually predict the isoelectric point of your protein using a pH gradient, right? So here you've got a variety of different proteins, and you can see you have the measured pH value on the y-axis, you have the pI value on the x-axis, and you see a very linear correlation. You have an R squared of 0.99, right? So it's a good way to figure out the pI of your protein. The other great thing about using a pH gradient is that you can use the same gradient for all of your methods, right? Basically, you can use a pH gradient for scouting. So here you've got two biosimilars, Biosimilar 1, Biosimilar 2. Using a pH gradient for both of them, same one, you get two different retention profiles. So it's just easier. And then finally, I think this is my last slide. We're looking at a biosimilar profile of charge variance here. So again, with cation exchange, your acidic variants come out here in the front. These are not going to be very well resolved because they elute early on in your run. You have your main peak, your main monoclonal antibody, and then your two terminal lysine variants come out in the basic region, right? And so these are a little bit better resolved. Okay, that's it. Basically, I talked about why biopharma is growing faster than pharma. 5 of the top 10 pharmaceuticals are monoclonal antibodies. It's projected to be a $140 billion industry by 2024. And then I talked about structure of mAbs, post-translational modifications that happen, and 3 main types of chromatography: size exclusion, reverse phase, and ion exchange. So if you guys have any questions, that is what I'm here for now.